So good evening, everyone. We are going to talk about techniques of pipetting or pipetting techniques. So for our objectives, it is to determine the importance and use of pipettes and also to identify the different qualifications of pipettes. To also know and practice on how to use the pipettes and able to know the proper pipette techniques. For our introduction, this will be discussed by Rashid Claire G. Mongoy.
Good evening, everyone. I I'm Rashid Claire Mongoy, and I'm one of the reporters of Group 3. So for the introduction, pipettes are essential in almost all of the laboratory procedure. Pipettes are used for obtaining particular measurement for liquids. Lacking in knowledge about proper pipetting technique might affect your overall experiment. It should be remembered that not all glassware are designed either to contain or to deliver. So before we discuss the different classification of pipettes, let us watch the video first for us to understand the topic.
Walay audio.
Gani, walay od.
From Step 1. Draw up the liquid to be measured. Hold the stem of the pipette between the thumb and the middle fingers of your dominant hand. Allow for the movement of the index finger on and off the top of the stem. Hold the pipette vertically through the rest of the steps. Place the tip of the pipette into the fluid to be measured. Make sure that the tip is far enough into the fluid to fill the pipette without drawing up air. Expel air from the pipette bulb and place the bulb firmly on the top of the pipette stem. Slowly release pressure on the bulb to draw liquid up the pipette past the calibration mark by about 1 centimeter. Maintain steady pressure to prevent fluid from contaminating the bulb. Remove the bulb and cover the top of the pipette with the index finger. Check for bubbles. If there are no bubbles, proceed. If there are bubbles, drain the liquid back into the beaker and try again. Step 2. Measure the liquid. Lift the pipette out of the liquid and wipe any excess liquid from the outside of the pipette without touching the bottom. Holding the pipette vertically, angle the beaker to place the tip of the pipette against the side. Do not allow the pipette to go back into the liquid. Raise the calibration line to eye level. Slightly release pressure from the index finger, allowing the liquid to flow at a slow and steady rate. Press down firmly with the index finger when the bottom of the meniscus is sitting on the calibration line. Step 3. Deliver the liquid. Move the reagent beaker out of the way and angle the labeled receiving vessel into position. The pipette remains vertical. The tip of the pipette touches the side of the receiving vessel. Raise the graduation line of the volume to be delivered to eye level. Release some pressure from the index finger to allow the liquid to flow at a slow and steady rate. Press down firmly with the index finger when the meniscus has reached the desired graduation. Repeat this step if you are measuring multiples of the same volume, raising the next graduation line to eye level for each delivery. Remember, do not drain to the tip. Bore pipettes are only calibrated to the last graduation line before the tip. Excess reagent in the tip must be disposed of in a waste container and not deposited back into the original container.
For the different classifications of pipettes, this will be discussed by Irene Parocha and Stephanie Villanueva.
So for the letter A, according to manner of calibration, first let's identify what is calibration. So calibration in its simplest terms is a process in which an instrument or piece of equipment's accuracy is compared with a known and proven standard. So number one, to deliver or TD. This type of calibration will dispense the exact volume indicated. The small amount left in the pipette tip should not be blown out. Water is used as the calibration medium. And for the number two, to contain or TC. To contain, pipettes will hold or contain particular volume, but does not dispense that exact volume. The exact volume can be delivered by repeated filling, then emptying the diluents or rinsing techniques. And for the number three, to blow out pipette. Similar to TD pipette, except that the exact volume is obtained when the last drop on the tip will be blown out. An etched ring or frosted ring indicates its calibration. And for number four, between two marks pipettes. Exact volume is calibrated to fill the volume between two calibrated points on the pipette. And for the next reporter.
Okay, good evening. To continue, for the according to graduation, we have number one, volumetric or transfer pipette. It transfers a known volume liquid. It is calibrated to deliver accurately a fixed volume of a dilute aqueous solution. And they include both volumetric Ostwald-Falling pipettes. They consist of cylindrical bulb joined at the both ends to the narrower glass tubing. So we have here Ostwald-Falling pipettes. For further knowledge, or Ostwald-Falling pipettes are a type of volumetric pipette with bulb near the delivery tip, and it is usually, or they are TD, or what we call to delivery, but are designed for use with more viscous fluids such as blood and serum. Moving on, for the number two, measuring or graduated pipe. They are calibrated to dispense different volumes. It has uniform graduations along its length, and there are two types of graduated pipettes. Number one, we have Moore pipette and serologic pipette. So what are the difference of these two? According to my research, a serological pipette is a graduated pipette in which the calibration marks extends all the way to the tip, whereas Moore pipette is a graduated pipette in which the calibration marks do not extend to the tip. So for the next slide, we have here for the last one is a micropipette. In microwork, the remaining volume is left in a pipette can cause significant error. For this reason, most micropipettes are calibrated to contain the stated volume rather than to deliver it. So this is the structure of a micropipette, and it is usually different with our previous pipette, which is the serological and Moore pipette. So next. Here, in figure number one, this is the right reading for our graduation mark, where it should be on our eye level, and this is what we call meniscus, the U shape there, and it is mentioned earlier by Irene Parocha. For the second figure, we have here Ostwald, volumetric, serologic, and graduated. And these are the difference of the two. I have mentioned earlier that a serologic pipette is its calibration mark extends all the way to the tip. So as you can see, it is shown there, the second pipette, that is the serologic. While the more is like the same in this first pipette. Moving on with the procedures to be reported by Mr. Magallanes.
For the materials, we need pipettes and pipette bulbs. Pipettes will be used, or it is an instrument in the laboratory that is being used to measure out and transfer small quantities of liquid. The pipette bulb, which is you can see here in the second figure, um, it is used to transport a measured volume of liquid. For the next slide is the procedures. So for the first step, you have to wear your PPEs for your own safety and prepare a proper pipette to be used. Third step is rinse the pipette with a small amount of the solution to remove any water film from the inside walls. To fill the pipette, compress the rubber pipette bulb and put over the top stem of the pipette. Insert for the fourth step is you have to insert the pipette tip into the liquid and slowly release the pressure on the pipette bulb. Allow the liquid level to rise above the calibration mark. Do not permit liquid to enter the rubber pipette bulb. For the fifth step is you have to remove the bulb and quickly fit your index over the stem. For the sixth step is allow the level of liquid to drop until the meniscus is exactly level with the calibration mark by adjusting the pressure of the index finger. Measure the volume by eye level. Touch for the seventh step, you have to touch off the hanging drop from the tip of the pipette. Or the tissues are not recommended because the paper fibers can draw liquid out of the pipette tip by capillary action. For the eighth step, to transfer the liquid to another container, simply release the finger pressure on the pipette stem and allow the liquid to drain freely. When finished, touch off the drop of liquid hanging on the tip, at the tip into the transferred liquid, but do not blow out any liquid remaining inside the tip of the pipette. The pipette has been calibrated to contain this last drop of liquid. For the next slide is for the conclusion, which will be reported by Mr. Gilo.
So for the conclusion, in this experiment, we learned about the different pipetting techniques and its different classifications of pipettes, like according to manner of calibration. Example for these are to deliver and to contain, and according to its graduation, because it is calibrated to deliver an accurately fixed volume. And we also learned that the micropipettes are calibrated to contain the stated volume. And we also learned that they are used for obtaining particular measurement for liquid, especially when you want it to be an exact amount.
So that's all for our report. God bless you all.